Picture a petri dish sitting overnight in a hospital lab. Everyone's gone home, the lights are off, nothing's supposed to be moving. Yet, by morning, those bacterial colonies have crawled across the plastic like tiny midnight hikers. No flagella, no swimming. So how on earth did they get from A to B? Here's the thing. On a smooth surface, think lab plastic or even the slick lining of your throat, bacteria can't just push off the ground the way we do. And if their propellers are disabled, the usual playbook is out the window. For decades, we boxed them into two neat categories, free swimmers or sticky squatters. Turns out that box is way too small. Today, we're going to unpack the surprising backup plans bacteria use when swimming isn't an option. First, we'll look at the chemistry-powered sailboat trick. Then, we'll meet a micro-machine that works like a winch. Finally, we'll zoom out to see why these hidden moves matter, from catheters to garden soil. Sound good? Let's dive in. Signpost 1. Chemistry in motion. On soft agar, a jelly that's mostly water, certain strains of E. coli and salmonella pull off a hands-off hustle. The cells chew through surface sugars and spit out tiny detergent-like molecules called biosurfactants. Those secretions create gradients in surface tension. Picture soap pulling grease across a sink. Here, the gradients tug the thin liquid film sideways. The fluid starts sliding, and the bacteria, essentially hitchhikers in that film, get carried along for free. Researchers proved it with fluorescent beads. Under the microscope, the flow isn't a random swirl, it's an organized fan. Fast lanes at the edge, slower traffic inside, almost like rush hour highways setting their own speed limits. Block the sugar processing enzymes, and the whole traffic pattern collapses. Change the moisture or the gel's stiffness, and the expanding ring reshapes itself. Chemistry, not motors, is in the driver's seat. Let that sink in for a second. Signpost 2. Mechanical muscle without muscles. Other microbes go full engineer. Meet Flavobacterium johnsoniae. Its outer surface is studded with adhesin proteins. Think of them as microscopic cleats on a shoe. Inside, a rotary gear meshes with a helical track. When the gear spins, those cleats march along the cell's surface. If a cleat grabs the ground, the whole cell jerks forward, exactly the way a rock climber hauls on a grip. Shift the internal gearing, and the bacterium can either power walk across smooth terrain or grunt its way through sticky goo. So, even when we knock out the classic flagella gene, these bugs just reach into a different toolbox. Signpost 3. Why it matters in the real world. A catheter in your bloodstream isn't a featureless tube. It's full of micro-scratches that trap moisture and nutrients. Those grooves become express lanes, guiding bacterial spread in one direction, while the rest of the surface stays quiet. In the mouth, plaque uses changes in saliva thickness to zip along braces or between teeth. Micron-small highways we'd never notice. Soil communities pull the same stunt between sand grains. A hair-thin water bridge suddenly links distant neighborhoods. Even inside the gut, patches of mucus with slightly different oxygen levels can channel certain microbes so that one spot gets overrun while a neighboring patch stays nearly pristine. Add it up, and you see why knocking out a single motility gene almost never stops colonization in the wild. Surfaces are landscapes, and bacteria are master cartographers. Looking ahead. We've mapped the microbial interstates, flagella, pili, the obvious stuff. The side streets are only now coming into view. New imaging tools that paint flows and forces in real time could tell us which routes pathogens choose and when. If we can redirect that traffic, block a shortcut here, open a detour there, we might steer entire communities instead of chasing rogue cells after the fact. So what does this mean for you and me? Better coatings on medical devices. Smarter dental treatments that shut down plaque's preferred lanes. Even agriculture that harnesses helpful flows to spread beneficial bugs faster than the harmful ones. Recap and take home. So here's what we covered today. 1. First, bacteria don't quit when their flagella are gone. They tap chemical gradients that literally pull them along like surfers on a tide. 2. Second, some species build internal winches, tiny mechanical systems that walk them forward cleat by cleat. 3. Third, the environment decides which backup plan wins. Catheters, teeth, soil, and guts each hand bacteria a different map, and they navigate accordingly. 4. Finally, if we can learn to redraw those maps, changing surface chemistry, moisture, or texture, we could slow infections, guide probiotics, and maybe even shape whole ecosystems. Remember, microbes aren't just passengers on the surfaces around us. They're active drivers, constantly opening new roads. The better we understand their hidden traffic system, the better chance we have to keep the good bugs cruising and the bad ones stuck in gridlock. Thanks for listening. If you enjoyed this episode, share it with a friend who still thinks bacteria just swim or sit still. And as always, stay curious.